Kevin Bowling received his PhD in molecular biology and biochemistry from the University of Alabama. He joined HudsonAlpha in 2009 and is utilizing a number of genomic techniques to better understand molecular pathologies that underlie neurological phenotypes. But what's really interesting is he is currently working on childhood genetic disorders, finding desperately needed answers for families who are struggling with undiagnosed diseases and disorders. Thank you and welcome.
Thank you. So I want to keep this really informal. So if you have a question while I'm talking, please feel free to chime in, and maybe we can turn this less into a presentation and more into a discussion. So as Stephanie mentioned, first of all, thank you guys for giving me the opportunity to talk to you about some of the cool things that are going on at HudsonAlpha. We're doing a lot of exciting work, and I am so happy to be here presenting about this work tonight. I'm going to talk about kind of a small facet of what HudsonAlpha is doing, although it's also a big part of what HudsonAlpha is doing, and that is looking at clinical childhood genetics. More specifically, I want to talk about a project that I've been helping to kind of drive over the past 2.5 years, and that is asking the question, can we get a genetic diagnosis for children who have a rare disorder? So I think most people in this room probably know about HudsonAlpha and what HudsonAlpha is, but let me answer that question first just to get us started. So HudsonAlpha is a nonprofit biotechnology institute. We're located in Cummings Research Park on 150 acres. The institute has 4 different missions. One of those missions is to do scientific research, make new discoveries, kind of advance the field. Another mission is we have a very far-reaching educational outreach program that for those of you who have taken Biotech 101 or 102, you already know about. Another mission is we kind of want to drive economic development and economic growth. And we do that by kind of being housing a number of different associate companies within HudsonAlpha that are all focused on biotechnology. And then the 4th mission is one that's brand new, and this is probably the one that you guys may know most about just because of the media storm that happened in the fall, and that is our clinical genomics program. So in October/November, we opened up a new clinic. That's the Smith Family Clinic for Genomic Medicine on campus, and we're now seeing patients at HudsonAlpha that have rare disease. So that's kind of what HudsonAlpha is about. Our first building opened its doors in 2008. At that time, we had 8 different associate companies, 3 faculty researchers, and 3 educational outreach members. So you can see we were pretty small when we opened. The picture on the right is currently our campus, and what you'll probably notice is McMillan Park with the DNA double helix sidewalk. Our goal is one day we'll have buildings all the way up and down McMillan Park. The building on the bottom left, this is our main building. This was HudsonAlpha Building 1. This is the building that opened in 2008. Across the street, you can see we have the Jackson Center, which is a conference center. And then on the other side of the main HudsonAlpha building, we now have Building 3. And this is the house to an associate company and also to the Smith Family Clinic for Genomic Medicine. One of the things that HudsonAlpha is about is growth, and we're always constantly growing. We're always kind of raising the bar for ourselves. And you can see 7 years later, so 2015/16, we now have more than 190 people that are on the nonprofit side of the building doing research. We now house 28 associate companies, which employ more than 460 people. We have 15 faculty researchers, and we have 15 individuals in our educational outreach program. So we've grown a lot over the past 7 years. Now, obviously, one of the mission that I reside in at HudsonAlpha is scientific research. And this is, again, one of the main missions of HudsonAlpha is to advance the field in terms of science. We study a lot of different things at HudsonAlpha. We cross many different boundaries, many different disciplines, and this includes things like autoimmune disorders, diabetes, obviously childhood disorders. We do a lot of studies in cancer, and we also study lots of neurological disorders like schizophrenia, ALS, Parkinson's disease. And then we also have a strong interest in looking at plants that may be future biofuels, or also plants that may eventually feed the world. The one thing that all of these disciplines have in common is that we use DNA sequencing kind of to drive our research. And the thing that HudsonAlpha is focused on is doing DNA sequencing faster, cheaper, and better. We always strive to have the best technology in our building here in Huntsville, Alabama. And right now, the sequencing techniques and the sequencing technology that we're doing is some of the best in the world. And so just to kind of put things in perspective, this machine that I'm showing on the screen, this is called a HiSeq X. Okay, we have 10 of these machines within our building. Again, the best, the fastest, the cheapest sequencing that you can get. The first human genome was sequenced in about 13 years. The draft was released in 2000. It cost about $3 billion. Okay? Now at HudsonAlpha, in 3 days, using our technology, we can sequence 80 genomes. And obviously, it doesn't cost $3 billion per genome, right? So you can just see the improvements that's been made in technology. And we actually, take advantage of this technology so that we can carry out cutting-edge research. So I want to tell you today about our program in pediatric genomics. And this is something that's really dear to my heart. I've been working on this project really closely with a lot of our genetic counselors that are sitting at this table here over the past 2.5 years. And I want to kind of tell you how the project is going, where we came from, where we've been, and share with you a few success stories so that you can kind of see how things are going. So I mentioned to you that kind of the bread and butter at HudsonAlpha is DNA sequencing. Okay, and so the idea behind the Pediatric Genomics Project is that we're going to use DNA sequencing to genetically diagnose children who have a rare childhood disorder. And sometimes you'll hear us say rare childhood genetic disorder, and that's because we believe that most of these disorders have a genetic basis. Okay? And this project is funded by the National Institutes of Health. We kind of work under an umbrella project called CSER, and that is Clinical Sequencing Exploratory Research. And the goal of this consortium is just for us to figure out how to take genetic sequencing into the clinic. So genetic sequencing of human genomes has been done for years now, but it's been done more in a research setting. So the idea that or kind of what we're working toward is making that part of medicine, right? And so we're doing that through our study, and then of course our Clinic for Genomic Medicine is doing the same thing. And for this particular project, we're teaming up with scientists and clinicians at the University of Louisville and also UAB through Children's of Alabama. And the question that we're essentially asking is, if we have a child who has some type of rare disorder, can we go in and sequence their DNA and identify the DNA mutation that is resulting in that disorder, right? So essentially giving them a genetic diagnosis. And I'll talk more in a second about just some of the hardships that families who have a child with a rare disorder, some of the hardships that they face because they're essentially on a diagnostic odyssey, right? They want to know what's going on with the child, and so often the diagnosis is inaccurate or the diagnosis is just unknown. So we're hoping by doing this project we can greatly help these families. This project is funded for 4 years currently. We're hoping for an extension so that we can do more studies like this in the future. But we are in year 3 of this project at this point. Over the 4-year span, we hope to sequence 450 families, so that means 450 children and their parents, and I'll explain more about that in a second. And so by the end of this story, we will have sequenced almost 1,400 individuals. Okay? So you may be asking yourself, wait, what is a rare childhood genetic disorder? Okay? And I call these rare, but the fact is if you take all children that have a rare disorder and band them together, about 1% to 2% of all kids are affected, right? So rare disorders are not really that rare. And when I talk about disorders, I'm talking about kids who are born with intellectual disability, so they have cognition impairment, developmental delay, maybe the pediatrician says they're not meeting their developmental milestones, children who have congenital heart defects, craniofacial and skeletal abnormalities, severe autism in some cases, or then some kids will also experience severe seizures. Okay, so this is the category of children that we're interested in helping, and you can imagine that these phenotypes or symptoms or this disorder is really severe, and we are pretty sure that there is some genetic cause that is just waiting to be found. Now, I already mentioned a little bit about the hardships that some of these families face. But there are a lot of challenges when you're trying to diagnose a child who has a rare disorder. For number one, they may be the only child that has this particular disorder in the world. So when they go into a specialist or a pediatrician, the specialist may have never seen anything like this before, right? So that makes things tough for diagnosis. But you can also imagine that if you have inaccurate, or if you have a diagnosis that is that's essentially unknown, that many problems can arise. So one example is you can have years of expensive, invasive, and simply unnecessary testing. And this was what I was talking about when I said there's kind of a diagnostic odyssey, right? Lots of different medical tests to figure out what's going on with my child, but you never get an answer. The other hardship is that it complicates treatment decisions, right? So you can imagine a physician who is seeing someone that has a rare disorder, and they're trying to figure out, how am I going to manage these symptoms? How can I treat this child? And there's simply no answer. It's very complicated. One thing that kind of touches me is that a lot of times parents will feel guilty about the situation, right? So they have a child who has a rare disorder, and there's always these questions of, is it something that I did? And I'll hit on this again at the end of the presentation. But parents, they have a lot of guilt, and they also feel like their child's health is literally out of their control, right? It's just kind of spinning out of control, and that's not easy to deal with. And then you can imagine also, this simply slows research in developing new therapies. If we don't understand the problem, we simply cannot fix it. So just listing all of those things out, you can see that us finding the root genetic cause of all of these different disorders is essential, right? Essential so that we can counsel the patient and their families, so that we can treat the families, and also that hopefully we can improve treatment for future generations of children who have a childhood genetic disorder. So let me just kind of lay out exactly what we're doing in our study, and then we'll talk about some results. So first of all, I've already mentioned we're recruiting 450 families, right? Mostly trios. When I say trios, I mean we are sequencing the DNA of a child, and then we're sequencing both parents. And I'll talk about that in an upcoming slide, and you'll understand why. And of course, we're enrolling children who are affected by some childhood disorder that need a diagnosis. And then we're conducting DNA sequencing. So we're sequencing the whole genome. And when I say genome, I just mean all of the DNA within a cell. Okay? We're sequencing all of DNA to ask, can we identify the genetic mutation that we believe is causative, okay, that is actually relevant to the child's disorder. And if we can, then we will return that variant or that DNA mutation to the families. And hopefully by doing that, we can say, we've identified the mutation, this is the disorder that we believe your child has. And then finally, I mentioned that we're kind of working under this umbrella of clinical sequencing exploratory research, right? Keyword exploratory. You know, genomics has not been moving into the clinic for a long time. Genomics has not been part of medicine, but for the past few years, right? So this is still exploratory, so we understand, we're cognizant that returning results like this to a family could cause them some strife, right? We know that there can be some negative impacts of doing this type of medicine. So we're also carrying out studies where we're looking at kind of the social and the ethical implications of doing this type of research, and even basically practicing this type of medicine. Okay, so I mentioned that we enroll the child who's affected, and we enroll two parents, right? The parents of each child that are unaffected. And the reason we do that is a really simple one. So you can imagine, or we know that, you know, if we think about my DNA, I got half of my DNA from mom, and I got half of my DNA from dad, right? So if I have a disorder that neither of my parents have, then there must be something different in my DNA. Okay, there must be some new mutation in me that is resulting in the symptoms that I'm experiencing. Okay, so that's why we're sequencing child, mom, and dad, because basically by a procedure of deduction, we're saying, what does the child have? What is specific to the child that mom and dad don't? And that may be what's causal. Okay, so this is how we're actually carrying out our experiment. So this project started in 2013. I mentioned that we are about 2.5 years into the project. We began enrolling our first patients in June 2013. Right now, we have enrolled 283 families into our study. Most of these families are trios, again, meaning we have child, mom, and dad. But we are also we have been enrolling children who have none or only one biological parent available for study. And the reason we're doing that is because we started to realize that there are a lot of affected children who maybe dad is out of the picture or maybe they are adopted, and we wanted them to be able to benefit from our study as well. So now we're enrolling any child who is affected regardless of parent participation. Interestingly, we have two families that have affected identical twins. We've actually diagnosed one set of identical twins. And then we have multiple families who have more than one affected child. Okay, so you can imagine where there's a child and then they have a brother and a sister who's also affected. And these cases can be a little bit more difficult. I think that the genetic architecture underlying the disease is a little bit more complex, so we're not as successful at diagnosing these families. But nonetheless, if we don't analyze these types of families, we're not going to learn how to interpret that data later. In terms of sequencing and analysis, of the 283 families that we've enrolled, 246 of these families have been sequenced, had their whole genome sequenced, and have been analyzed. And of those 246, 201 have actually received results in the clinic. So they know whether or not they got a diagnosis from our genetic test. So you may be asking yourself now, okay, Kevin, you're talking about all these families that are enrolled. Do they all get a diagnosis? Okay? The quick answer is no, but let me tell you kind of what our results have been to date. So again, 246 families. The pie chart to the right is showing you percent families. Okay? So you'll see that 27% of our families, so that's 67 children in total, have got a genetic diagnosis, right? We've found a DNA mutation in them that we actually know what disease it causes or that it leads to, right? And I've also listed a number of different syndromes here, which I'm not going to go through this list, but you can see that we've diagnosed children with many different rare disorders. And we continue, for those that don't get a diagnosis, to go back and reanalyze to try to give them something that's useful. 15% of families receive an uncertain significant genetic finding. And what I mean there is, imagine that we find something in the genome of an affected child. It's something that we think may potentially lead to their disease, but we're not 100% sure. We will label that as a variant of uncertain significance, and we will go back and follow up on that variant because some of those actually may be diagnostic in the future. Okay, so we follow up on those and make sure that if new information comes out, we may be able to reclassify that as a diagnosis. And as I mentioned, there's a number of children in our study that do not get a genetic diagnosis. Here you can see 58% actually receive a negative result. This is primarily because we don't completely understand the genome yet. So we can go in and identify lots of different genetic changes that are occurring. That does not mean that we can interpret them, right? So for the most part, a lot of this is just that we don't understand the genome. Sometimes we find mutations in genes that we don't understand the function of those genes. But as more and more science is being done, we're learning more and more every day. And so we're constantly going back and reanalyzing these families in hopes of one day being able to give them a diagnosis. Okay, so that's the results of our study thus far. I want to take you through a case study now. A lot of you guys are probably not looking at pedigrees all day like we are, but the diagram that you see here, this is called a pedigree. This is actually a really simple one. We're just showing mom, dad, and child here. In the top right, the open circle, this is the 36-year-old mom. Okay, you can see that she has asthma. Otherwise, she's alive and well. On the left, this is a 47-year-old male who is the father of the affected child. He's the open square. He has complications related to diabetes. Otherwise, he's alive and well. And so you can see that both mom and dad have no phenotypes or no symptoms that may be indicative of what the child has. And so therefore, we would call mom and dad unaffected for these purposes. The affected child, or the child who has the rare disorder here, is a 3-year-old female. Her clinical presentation is hypotonia. So she has low muscle mass, low muscle tone, really weak muscles. She has severe intellectual disability, so really heavily impaired cognition, microcephaly, small head, speech delay, exhibits seizures, Brain MRI says that her brain structure is abnormal, and then she also has problems with visual maturation. So a number of different symptoms, and you can see some of those fall into that spectrum I showed early on when I was describing childhood disorders. But you can imagine getting a clinical report from our genetic counselors and they being different for each child, right? There's a number of different symptoms that can fall within these categories. So we sequence mom, dad, and child, and we ask the question, what is different in the child? And this is a case, obviously I'm going to show you a successful case, right? But this is a case where we did have a diagnosis. And so in this family, what we found was a de novo variant. Okay, so DNA sequence revealed a de novo variant. This is a new mutation in the child, meaning it wasn't inherited from mom or it wasn't inherited from dad. And this variant is rare. And when I say rare, I mean it is not in any public database that's out there that has human genetic information in it. So it could be that this child in our study is the only child in the world that has this DNA mutation. Okay? And interestingly, this mutation falls on a gene called FOXG1. And FOXG1 is a gene that we know a lot about. It's actually, when you have mutations in this gene, it leads to a version of Rett syndrome, and I won't go into the details of Rett syndrome, but let me just say that the phenotype that our child has matches up really well with this disorder, okay? So this is a variant that will be returned to the family as pathogenic. We will say, you have a diagnosis, your child has this version of Rett syndrome, okay? Now, when we were doing this case, it was interesting because the genetic counselors put in a lot of clinical notes that the analysts can go in and read so we can understand what the symptoms are, and we can also understand some things about family structure. And I just wanted to read this clinical note from you because this really touched me, and I think it will you too, and it just kind of shows the importance of doing these types of studies. And it's easy for us to stand here and say, you know, oh, we're able to diagnose 27% of kids. But if you are one family in this study that's received a diagnosis, then you can truly understand how this study affects people. So this is the clinical note. I'm reading straight from this is exactly what was in our database. Both parents are deeply desiring a diagnosis and more information. Dad was tearful as he said he wanted to know what his child has. Mom verbalized that she wants a diagnosis, but also was scared that the diagnosis will be bad. Dad responded saying that he wants to know regardless, even if it means his child only has 6 months to live. The dad also talked about how not knowing is worse than finding out any kind of poor prognosis. So you can just read this comment and you can tell that this family has been through this horrific diagnostic odyssey, right? Lots of different genetic tests, lots of different medical tests, and never an answer. And so now this family has an answer, and we can go back to the clinic and say, your child has Rett syndrome, and they can start seeing a Rett specialist in Birmingham. They can, meet up with other families that have children that are suffering from the same type of disorder, and this can give them some peace and also dissipate some guilt that, hey, this is a new mutation. This is not something that they inherited from mom or dad, and this is not something that mom did during pregnancy, right? So I'll tell you one more quick story, and then I'll wrap up. So this is a story about a 13-year-old female who we sequenced. We sequenced the affected child, and we sequenced mom and dad. We found a new mutation in a gene called TCF4, again, not inherited from mom or dad. This particular gene, when mutated, has been well described in the scientific literature, and it leads to a syndrome called Pitt-Hopkins. Okay. The mom from this study was ecstatic to get this result because now, and this is a really outgoing person, now she is involved in Pitt-Hopkins support groups. She's meeting up with the other families that have children that are affected by the same disorder. She says that she now has a newfound hope about the future for her family and her child. And she also says that the guilt that was associated with this disorder for so long has been dissipated by this finding because it's a new mutation, right? Completely out of her control. I wanted to show you a quote that, again, was given to our genetic counselors at return of results for this family. The mom said, "I've always been told that my daughter's disorder was not because of anything that I did, but until you are told what the cause is, you always wonder if you're to blame." So again, you can imagine the relief that this mom had to get the result, not only the dissipation of guilt, but now they can start seeing specialists and they can think, what the prognosis of the child may be based on other Pitt-Hopkins syndrome, and they can get a lot out of just, getting a result from this study. Okay, so I think that's all I have. Of course, the pointer's going to stop on the very last slide. Anyways, the last slide is just a quick acknowledgement to say there are a lot of people involved in this research. There's actually a Caesar table over here that has a lot of people that are involved in the project, and well, there we go. Yeah. Anyway, thanks to everybody that's involved, and thank you guys for your attention.